Today, me and Brian are going to be talking about DNA and RNA. We're going to be talking about the overview, structure, and DNA replication.
We're going to go over nucleic acids, DNA and RNA structure, and DNA replication, and wrap up. We'll wrap up at the end.
All right, so nucleic acid overview. Nucleic acids are monomers or macromolecules which are essential to life. Macromolecules are a type of molecule which contains a large number of atoms. Nucleic acids are polymers, which are made up of nucleotides, which are the monomers. There are two main types of nucleic acids: deoxyribonucleic acid, and also known as DNA, and ribonucleic acid, also known as RNA.
So DNA dumbed down. So first of all, its pentyl sugar is deoxyribose. Its structure is a double helix. Its role involves storing long-term generic information. It's permanent and usually does not go under significant changes. It carries a genetic code and is usually stored as chromosomes inside the nucleus. RNA dumbed down. So unlike DNA, its pentyl sugar is ribose. It forms a single helix, unlike DNA's double helix. It has many different functions, such as gene regulation, temporary genetic storage and transport of information, and assisting in many processes such as cellular respiration and protein synthesis. All RNA is made from DNA transcription, and some viruses use RNA to carry genetic information.
There are three parts of a nucleotide. First of all, there's the phosphate group, which is a phosphate. It's a charged group containing PO4. Then there's a pentose sugar, which differs from DNA and RNA. A pentose sugar is a sugar containing 5 carbons. And then there's a nitrogenous base, which is one of the 4 of G, C, A, T, or U. They're the nitrogenous bases. They're purines and pyridamines, I think. Purines are have 2 carbon nitrogen rings, and pyridamines have 1 carbon nitrogen ring. DNA and RNA both have 4 nitrogenous bases, 2 purines and 2 pyridamines. DNA has adenine, guanine, I don't know how to say it, cytosine, and thymine. RNA has adenine, guanine, cytosine, and uracil. Here are the pictures, I guess, of the 4 bases. Did I skip one? Base pairing. Sorry. Did I? Purines must pair together with a pyridamine. For DNA, it's AT and GC. For RNA, it's AU and GC. So for RNA, uracil replaces the T. Though RNA is single-stranded, base pairing is necessary sometimes. Hydrogen bonds hold together the nitrogenous bases. AT and AU both have 2 hydrogen bonds, as you can see in the diagram. GC has 3 hydrogen bonds, as you can see in the diagram again. Phosphodiester bonds. The OH on carbon 3 with, if you could see, actually, I don't have anything at that point. And the OH from the phosphate group combined. The OH is combined under a dehydration synthesis reaction, also called a condensation reaction, leaving water as a byproduct. This links together the phosphate group and the pentose sugar. These bonds form the phosphate sugar backbone of DNA and RNA. The direction of strands. DNA is anti-parallel. This means that the strands run in opposite direction of one another. So if one strand is 3' to 5', the other is 5' to 3'. A DNA strand has 2 ends or caps, a 3' cap and a 5' cap. The 5' cap is the phosphate group. This is because the phosphate group is attached to the 5th carbon on the pentose sugar. The 3 cap is the OH group. This is because the hydroxide or OH group is attached to the 3rd carbon. DNA and RNA, structural differences. Clearly, one is double-stranded and the other is single-stranded. However, this is because DNA uses deoxyribose, while RNA uses ribose. The 2nd carbon on deoxyribose has a hydrogen instead of a hydroxide group. This allows it to bond via dehydration synthesis in a way ribose cannot.
DNA replication. Sorry.
Oh, shoot.
DNA replication. So DNA replicates itself semi-conservatively. This means that the 2 new strands of DNA have half of the original DNA strand, as you can see in this diagram. DNA splits and has DNA polymerase attached to according bases because each base has a corresponding pair. And this is a very simplified process of what actually happens. We'll be going into the more complex explanation afterwards. But first and foremost, it's important to understand that DNA replicates itself semi-conservatively. And yeah, nitrogenous bases only have 1 associated paired base, so DNA synthesis can code off either strand. And DNA replication involves quite a few enzymes. So if you ever see a chemical that ends in A's, like A, S, E, you can assume it's going to be an enzyme like topoisomerase or lactase, which like that's lactose. Milk is great. So to elaborate on the simple law that was presented earlier, DNA is far more DNA replication is far more complicated than that. Firstly, DNA is unraveled through DNA helicase, an enzyme, as you stated earlier. Topoisomerase ensures that the DNA ahead of the split is closed correctly by cutting the phosphodiester bonds between nucleotides. Due to the anti-parallel nature of DNA, there will be a leading and a lagging strand, which is important to know in replication. This is because DNA polymerase can only go from 3' to 5' on the template strand. It cannot go the other way, or else this would be a lot more simple. Next. So the leading strand will have DNA polymerase 3 synthesize DNA in a continuous manner, approaching the replication fork. And the replication fork is where helicase splits the DNA into two half strands. The lagging strand will have broken synthesis, with DNA primase making RNA primers, resulting in short strands of DNA called Okazaki fragments, as DNA polymerase 3 cannot continuously add like it does in the leading strand. So an analogy I like to use for this is because while the leading strand, imagine you walking on a treadmill. You can just continuously do it, right? Walking like support facing the area on the treadmill. Whereas on the lagging strand, imagine walking in the direction the treadmill is pushing. Every few seconds, you end up off the treadmill and to get back on. And this place where you get on would be analogous to the RNA primer. And that's why on the lagging strand, it cannot synthesize in a continuous fashion. As you see, it has to go back and make another Okazaki fragments. Eventually, I should also clarify that RNA primers are RNA nucleotides. Eventually, these RNA primers are then removed and replaced with DNA nucleotides by DNA polymerase 1, and they're joined together through the phosphate backbones by DNA ligase. After replication is complete, DNA automatically lines itself back into a double helix, and DNA polymerases are usually capable of checking the strand they just made from mistakes. This will ensure that replication has gone through without any issues, which is integral because obviously DNA holds all the genetic information. This is necessary to fix common issues such as base mismatch, and DNA can be checked for repair at many points afterwards, such as when the cell is prepared to divide in the cell cycle. That's it. Okay, so this is an overview of the reproduction enzymes. You probably can't see it because of that overlay. So DNA polymerase comes in many types, but the ones that are important for today's lesson are DNA polymerase 3, which attaches nucleotides to replicate, and DNA polymerase 1, which removes RNA primers and replaces them with DNA. Topoisomerase cuts through phosphate backbone to fix coiling issues of the DNA ahead of the replication fork. If you don't remember, the replication fork is where the two DNA half strands separate. DNA primase is a type of RNA polymerase, and it forms RNA primers on the leading strand once and many times on the lagging strand to start off the RNA polymer. RNA polymerase 3, because DNA polymerase 3 requires an RNA primer to start forming DNA. DNA ligase attaches DNA fragments together through phosphodiester bond formation, which it catalyzes. And finally, helicase unwinds DNA and breaks base pairs with hydrogen bonds. Okay, so I'll wrap up. If you don't remember stuff from the beginning of the presentation, there's the differences between DNA and RNA. So for function, DNA provides the long-term storage of genetic information, whereas for RNA provides information transfer, protein synthesis, and gene regulation. This is because RNA is far less stable than DNA and cannot be used for long-term storage. Structurally, DNA forms a double helix, whereas RNA forms a single helix. For composition, the base pairs used are adenine, guanine, cytosine, and thymine bases. And RNA uses the exact same except for thymine, which is replaced with uracil. And finally, for propagation, while DNA self-replicates, RNA is only formed from DNA when needed for cell's functions.
All right. I guess that concludes it. We'll be doing a Kahoot as a review. Today's lesson was kind of short because we got lazy.
No,
and
it was not because we're lazy. It was because we took it upon ourselves to give you guys some extra life.
And it was just
stop making us look bad.
Okay, and it was just a basic rundown of DNA and RNA. Okay, so are we doing a Kahoot?
Yeah, of course we're doing it.
How do I do this?
Yeah, I told you.
Brian, you want to come?
Huh? No, this is that will
How do I play? Hey, you want to make the Kahoot? Is it classic? Sure. All right. If you guys want to join the Kahoot, I don't really care.
Oh yes. Oh, what?
What are you doing? Oh my god. Hey, are we gonna do? I think our lesson was too short, right?
No.
I think I spoke too fast.
This is gonna take up 15 minutes. If I was gonna show them a video of DNA replication, we have no time.
Oh, really?
Yeah.
First, and then the Kahoot will get 3 pennies, second, step 2, and then become third to get 1. Then also from last week, I think it was Mia and Atisa and Bala.
I think we didn't cover enough because we cut out a bunch of information, so we thought it'd be cool.
Oh. No, I
We're gonna offer the good or I'm playing with the food.
Why? Come on, I can offer it.
I'm in ninth place. Holy shit.
Polite crowd. I'll move. Laughing, you're literally ball out of like we all know.
I don't remember
Whoa, kind of good. Wait, okay. You have been paying attention. Multi-select, so spicy.
Oh, this was easy.
Fucking trash.
Wait, how do I do
Oh, you got selected.
Wait.
Oh, you're what? You're so
I'm
bad.
second. Hey,
Stop tryharding. We're not giving
wait,
you more
I didn't
candy.
see it.
No, stop tryharding. Right, like we're gonna. Okay.
Michael, stop.
Okay, give me your phone.
Stop. No, stop.
Whoa.